Okay, we could get started. Welcome, everybody. This is the Musella Foundation's Brain Tumor Awareness Month webinar series. Our first talk is about effective treatments of gliomas. Can we deliver? And our two special guest speakers are Michael Volkelbaum and Anne-Marie Carbonell. You can see their affiliations right on the screen. Take it away.
All right, thanks, Al. Really appreciate the opportunity to be the first talk for Brain Tumor Awareness Month. And thank you for everything that you have been doing over, I think we've known each other for the past 22 years. And really all the work that you're doing to help patients and their families find better ways to treat this terrible disease. And so it's a pleasure to be able to talk about what Dr. Carbonell and I have been working on to try to advance our ability to take care of patients. As is usually the case, I have some disclosures that I need to share with you all, just to make it clear that anything that we do is a partnership between academia and industry, and these are some of the companies I work with. Dr. Carbonell, you want to?
Yes, sure. Likewise. Hi, everybody. And thank you also for Al for inviting me to be part of this. As the CEO of OncoSynergy, I have a direct equity interest in the company. And that luckily is all I have to disclose. So Mike, take it away.
All right, thanks. So, many of the slides that are going to be included in this talk are ones that I give at professional medical meetings, scientific meetings. And I always start with this slide because I think that we have to, as a field, understand the context. And hopefully this will help you all as patients and family members understand the context. And that is, it's still a very bad disease. Gliomas in general and glioblastoma, the outcomes are still not great. I mean, certainly we've made progress, but not the kind of progress we'd like to make. And the standard therapeutic modalities remain the same today as they've been in the past over decades. Surgery, radiotherapy, cytotoxic chemotherapies, things like Temodar. And then we have this new treatment, alternating electrical fields, that have made what I would consider to be an incremental advance, but not a revolutionary one. And the only approved therapeutics in the condition of recurrent GBM actually don't extend survival. And we've had many advances, and we talk about these quite a bit at our meetings, but really these have been understanding the biology and subclassification of these tumors, not so much in therapy. These are important advances. The more we understand the disease, the more likely we can find its weaknesses. And there have been many, many different types of treatments that have been investigated over the past few decades. A little bit of radiation works, let's try more. Let's try delivering radiation right into the resection cavity, that's brachytherapy. Let's try using very focused radiation instead of the diffuse radiation, that's radiosurgery. We've tried many biologics, those are the exciting new therapies that have worked in so many different cancers, vaccines, other immunotherapies, antibodies, when given systemically. They haven't worked so far, and many other classes of drugs. Signal transduction inhibitors, which are referred to as small molecule drugs, or other types of cytotoxic or DNA damaging chemotherapies. Many of trials, lots of promising leads, lots of excitement around them. Unfortunately, they have not yet translated into really the kind of survival we'd like to see. And this gives you an idea of what that timeline looks like. And these are really all of the approved agents, directly approved for glioblastoma. The nitrosoureas are the original chemotherapies. And then we had Gliadel, which was one of those nitrosoureas put into a dissolvable matrix. And then temozolomide came along. We know that as Temodar. And that started to increase survival more than these other drugs had before. Bevacizumab was approved based upon just seeing responses on an MRI, and I'll come back to that. And then we have the Novo TTF, or the tumor treating fields. And so in the end, we have some cytotoxic chemotherapies that were successful agents and many failed approaches, as I mentioned. Now, this bevacizumab, actually, when we looked at survival, whether or not it improved survival, unfortunately, it didn't show that. So again, this really comes back to so far, we've seen success from very dirty drugs. We've learned a lot about the tumors. Now we have to translate that into better treatment. And I think one of the things I've learned from this disease is to understand the different parts of the tumor. So there's a solid tumor component, and in this figure, it's referred to as zone 1. That's where you have really nothing but tumor cells, and you have some breakdown of what's called the blood-brain barrier that keeps most drugs out. It's what we also refer to as the enhancing tumor, contrast-enhancing tumor. And then there's another zone, or actually two more that we can describe around that. One is immediately outside of that, maybe about a 1-inch rim around it. And you'll still see obvious tumor cells there, but they're now behind the blood-brain barrier. This is what we refer to as non-enhancing tumor or tumor-infiltrated brain. And then there's a third zone, much farther away, where you may occasionally find tumor cells. And we have a number of studies from the laboratory that have shown that the cells actually look different biologically, the ones that are in the solid tumor versus the ones that are infiltrating. But the most important point is that when we are talking about delivering therapy, most of the studies we focused on when we look before getting to patients focuses on just zone 1, the solid tumor. Well, we already have an effective treatment for zone 1, that's surgery. We take that part out. What we're treating with all of these other therapies, the chemotherapy, the radiotherapy, is zone 2 and perhaps zone 3 as well. That's really what they target. That's what we need to be focusing on. It's not what we focus on quite so much in our field, unfortunately. So some of the factors limiting progress in our disease, well, it's infiltrative. As I just showed you, we have those other zones outside of the solid tumor. And when we talk about a maximal safe surgery, usually we're talking about removing that enhancing part, the zone 1 part. And it does provide a survival benefit, but it's not a cure, not alone. We actually have limited technology for identifying areas that contain neoplastic cells outside of that solid part of the tumor. And then these tumor cells can be very different from each other. Some cancers where we've seen some great progress are referred to as just single gene cancers. One gene was enough, one mutation was enough to push it into a cancer. And if you block what that mutation does, the cancer dies. That's not the case with GBM or gliomas in general. They're much more complex than that. And there's some implications for this infiltration. As I said, we can't call surgery alone a curative procedure for most of these tumors. We have looked at the question of going further, doing what's called a supramaximal resection, but so far, there are certainly successes in very limited tumors. If someone has a very, very tiny tumor that's in an area of brain that doesn't have a clearly described function, we could be pretty generous about what we take out there, and that probably helps. But that's not the way most of these tumors end up presenting themselves or growing. And so that's why that has not been as convincing of an approach. Ultimately, I think it comes down to the fact that most therapeutics can't get into the brain. And this has been shown over and over by the folks who make drugs, is that most of them don't get in the brain. And in fact, most of the drug makers don't want them to get in the brain because they don't want to have side effects associated with them. And so that's a problem when we think about having to give therapeutics systemically to get into the brain to fight cancer. But this has not been examined as carefully as it really needs to be examined. And I'll show some examples of that. And in fact, our clinical trials, which are designed to be very optimistic, sometimes don't ask that question either. So if we're to have progress in our field, we have to learn from our failures. And I don't think we've been doing a very good job about that. And a very smart man said, that doesn't work. We have to be smarter. So one of the questions I've asked in my career is our failed trials, are they a failure to understand the tumor biology or a failure to get the drugs or therapies to where they need to be? Now, starting with drugs that are given systemically, orally or IV, there's a claim that's made all the time. We say, originally the language was it crosses the blood-brain barrier. Now we use the claim brain penetrant. It's made very frequently, but what is the evidence? Well, here's some of the early studies looking at some of the really exciting new targeted therapies. And this was a paper from 2010. And you can see that the authors said that this drug efficiently crosses the blood-brain barrier in animal models. Okay, so I'm not going to go through too much data in this talk, but I think there's some things that will be self-evident. Okay, and I want you to focus on this part of the results. So this BAT is brain around tumor. So they actually implanted a tumor in a mouse, let it grow, and then treated it with this drug, which is supposedly crossing the blood-brain barrier efficiently. This is the drug level in that tumor. That tumor has a defective blood-brain barrier. It's same thing as an enhancing, contrast enhancing tumor. And the drug does in fact get into the tumor. This OH is opposite hemisphere. That's the part of the brain that doesn't have a tumor in it. And this is the brain around tumor. And you can see there's almost no drug in there. These are the parts that do not light up with contrast. They have an intact blood-brain barrier. This part does not. So does this really show that the drug crosses the blood-brain barrier efficiently? No, it doesn't. And that is one of many drugs that has failed in clinical trial. Here's another drug that was used in clinical trials. And this was an interesting approach in that the physicians here gave the drug to the patients before surgery, which I think is a very important principle here. They gave it before surgery, took out the tissue, looked in the tissue to see, does the drug really get there? Now remember, this is taking out enhancing tumors. So at least they're asking the question, does it even get into that part of the tumor where the drug can get in? And here are the amounts of drug that they saw in the tissue. And they said, yes, it does get in there, okay? But here's the question they didn't ask. How much drug do you need to actually cause the tumor cells to die? And other studies suggested that it was somewhere over here, much higher levels than what they were seeing in the tumor. And this does not address the issue of the infiltrated brain, where the blood-brain barrier is intact, and this drug probably didn't get in at all. So these are the challenges we have. So this is some work that I did early in my career. When I was at the Cleveland Clinic, we actually set up a laboratory to start looking sequentially at different drugs that were of interest at the time. We used this model. This is actually a rat that has a brain tumor in it. Here is an MRI showing you the tumor. And we would sample, we would give the drug that we were interested to the rat. We would then sample the tumor, and we would sample the unaffected brain and ask how much of the drug really got in, and then ask,
Wait a second, we lost you, you froze. We'll just give him a minute. And Anne Marie, can you hear me?
Yeah, I can, Al.
If he's stuck, could you finish this part?
Yeah, if he gets stuck, I have some slides. I don't know. I know he updated.
Oh, there he is. Wait, did you just get him back?
See.
No.
He's going to rejoin probably.
He'll rejoin. Let's give him a minute. Technical problems.
The joys of Zoom, huh?
Yes.
My worst nightmare is being stuck on Zoom like that with like my face frozen in an awkward position. So you should have it to Mike first.
That's strange because I have you here twice for some reason.
I know.
Maybe he clicked your link.
It could be some people on my team have our shared invite, so that's what I would
Okay.
My credentials. This is when you're supposed to tell a good joke, Al.
don't want to get into trouble.
I know, me neither.
You have to be so careful what you say these days.
I'm gonna keep my mouth shut.
They take what you mean and turn it around and yell at you. Okay, you want to start sharing your part?
Yeah, I know unfortunately, he didn't want me to share some of the slides that are OncoSynergy back, but I can pull up a different deck that has, because the Moffitt, it's recorded and there's so he has the master file, but I can certainly share something, even though it's a little bit less impactful without Mike's introduction. But I'll pull.
If you want to wait for another 2 minutes, we can.
Yeah, but I'll get something ready just in case.
Actually, I could ask you some of the questions we have already. You're a neurosurgeon.
Yeah, go for it. I'm happy to help if I can. Some of them, as you know, with my position at OncoSynergy, I have to be careful with FDA regulations about certain things, but I'm happy to see if I can answer anything while we wait.
Okay, this is just a general neurosurgery question. He mentioned that taking out more tumor didn't necessarily help, but is there anything to show that maybe if we took a slightly larger margin, you would do somewhat better?
So there is some evidence that shows that maximal safe surgical resection is beneficial. So there's literature that suggests that if we get up to 98% of a tumor, that is beneficial. But going beyond that and taking a margin like we do in other cancers, like in skin cancer, they'll typically take a margin of healthy tissue. Unfortunately, that has not been something that's been able to be studied significantly because the brain is so delicate. So we're unable to typically take a margin, and there has not been the risk-benefit ratio favors not going in that approach.
Okay. Why has so many treatments failed phase 3 when they had such success in a phase 2 trial? That doesn't make sense to us.
That's a great question. A lot of trials, when they're, you look at when you go through the different steps of drug development, you're looking at different things. So in the earlier stages in phase 1, you're really looking at safety and oncology. In phase 2, your goal is to take the dose that you found in phase 1 and test that in the patient population and look to see if you can find a preliminary marker of what we call efficacy or successful treatment. Sometimes when those endpoints are typically radiographic measurements, so what we'll do is we'll look at tumor response rates on MRIs. The question is that going to ultimately translate into increased survival in phase 3? And sometimes it doesn't. And what Mike was going to talk about, hopefully he will be able to talk about it when he gets back, is that one of the things that pharmaceutical companies don't really take into account for is does the drug actually bind to the target like we think it does? As it actually, can we actually see that it has the effect? And so, that's one of the things that we're doing in OncoSynergy's trial with Dr. Vogelbaum at Moffitt is we're really trying to understand if the drug works at the biological level and if there's certain patients that benefit more or less. And that's really important to ultimately the success in phase 3. But unfortunately, especially in glioblastoma, we haven't seen that translation of the early success into later phase trials.
Okay. There was a lot of enthusiasm for actually like 20 or 30 different drugs that wound up failing. Do you think it'll be worth going back and trying them using CED to see if the higher concentrations would make a difference? Because they worked in the lab animals and the models, but in the people, they just didn't.
Yeah, that's a really good question. And, definitely that's something I think that some people are doing. So repurposing drugs and now studying them via convection enhanced delivery, where we can confirm that the drug is actually reaching its target. In terms of, that's definitely a possibility. I would like to hear Mike's opinion on that as well. He could give more because he has studied some, he's done this in his practice, but it's definitely, if we actually get the drug to the target at a higher concentration, we should hypothetically have improved success.
Okay, from a legal standpoint, are you allowed to take a drug that is approved and use it in convection enhanced delivery, or does that have to be only in a trial?
Okay, so the question from a legal standpoint, if a drug is approved, for instance, for
Or
systemic,
something else, right?
right? So we still have to test it via trial and open up and submit a actual application to the FDA because the safety profile is much different when you're delivering it directly to the brain. So there is a little bit of a shortcut since you have a wealth of data already about approved drugs. But if we're going to use this technique in a device, there are additional steps that have to be taken, such as biocompatibility of the drug and the device, which is something that has to be submitted to FDA, as well as some other specific trial related data, and then it can be run under a clinical trial.
Okay, let me see. That's about all the questions I had. Can you give him a quick call and just see if
yes,
he's having problems?
I will switch some OncoSynergy slides and we can talk about the trial.
Okay, so we'll just wait for a few minutes. I'll put my Jeopardy music on. Actually, I could talk a little bit about the upcoming webinars. So I was going to talk about that at the end. I could talk about it right now. So while we wait, I just want to tell you about the rest of our lineup for the Brain Tumor Awareness Month. Next week, our speaker canceled. So right now the spot's open. I asked the doctor to fill in and we'll find out soon. I'll let you know. But on May 16th, we have perfusion MRI of brain tumors and a phase 1 study of a new candidate for high-grade brain tumors. It's a new drug that sounds really exciting. It just started phase 1 trials, but it worked fantastically on animals. So we'll see if it gets through the blood-brain barrier into the brain at the right concentrations. Then on May 23rd, we have a new hope in treating metastatic brain tumors, recent advances and opportunities. It's from the Brain Metastasis Clinic at the MD Anderson Cancer Center. They say they have a few exciting new things. We haven't really talked much about metastatic brain tumors. Metastatic brain tumors are about 10 times more common than the primary tumors, and we've been ignoring them. So we're going to try to get a little bit more into metastatic brain tumors. And lastly, on May 30th, I'm going to be the main speaker along with a few of my co-workers, Erica Monteverde, Tim Stumilla, and William Hughes. We're going to talk about the Promising Pathway Act, Patient Navigation Program, and ClinWiki. Okay, Dr. Vogelbaum's back.
All right, let me share my slide again here.
Yeah.
Can you tell me where you think I cut off? I'm sorry about that now.
Now go back one.
Go back one. Sorry.
Go back another one. Go back another one.
Yeah, you're on the in vivo. It was slide 17 on the deck that you had sent me. I don't know if it's still it.
Okay.
There it is.
All right.
So start again at the beginning of this slide.
Yeah. So sorry about that.
No problem.
I'll continue from here. So so the point I was making is that we actually set up an operation to start looking carefully at whether or not new drugs, these exciting new drugs, would actually be able to get to their targets in the brain. Looking both at enhancing tumor and in areas of brain where there wasn't tumor. So this is actually an MRI of a rat in which we implanted a GBM tumor. And you can see the tumor lit up here. And this is the opposite hemisphere of the rat brain. And we gave the rats the drug that we were interested in. And then we asked the question, how much of that drug actually got into the tumor? How much got into the normal brain? and what did it do there? Did it do what it was supposed to do? So the first, this example actually that I'm showing here is of a drug that at the time was called OSI 774. And this is the way that drug companies often name their drugs when they're early stage. They give them a number. It actually turns out that this drug you would know today as Tarceva. It was before it actually came on the market, before it was approved for lung cancer. And we were interested in this drug because it targets EGFR, which we know is an important target in GBM. So we did this study, we asked those questions, and what did we find? We found that when you looked at the drug levels in the tumor-infiltrated brain and the part of brain that had no tumor whatsoever, there was almost no drug. We did see it in the tumor, though, in the enhancing tumor. And so what this said was, this is a drug that is not likely to work. Now, we published this in 2013, but we actually had recognized this earlier. And in fact, there were a number of clinical trials being done at the time with Tarceva that ultimately failed. Now, I wish we could have done this before the clinical trials. That would have been a lot better, because then maybe those patients could have gone onto a different trial. And these are things that we can and we should do on a routine basis. That is to ask the question, what is this promising new drug doing in the tumor? This is an example of a study that David Reardon and Patrick Wen and I did. You may know Drs. Reardon and Wen from Dana-Farber. So we did this study together and with a new mTOR inhibitor that was being developed for GBM. And the question again was, does this drug actually get to the tumor tissue? And so we designed what we called a perisurgical phase 1 trial. We asked the question, is it really getting? And we gave the drug before surgery and then took out the tumor and looked at what happened in the tumor. So in this study, the patients had had a prior resection for their GBM when they were first diagnosed, and we took those specimens. And these are typical histological specimens that have been stained for specific proteins. In this case, this is a protein called S6. It's actually a molecule that is important in mTOR-related effects, signaling effects. And this is P27, another important protein that mTOR's affects. And then we took our surgical specimens that were taken after the patient had been exposed to this new drug. We said, what happened? Well, for this S6 kinase, for that protein, it may not be as obvious as I would like it to be here, but it actually inhibited the signaling via this S6 kinase. So that said, oh yes, the drug is getting there, it's doing what we want. Unfortunately for P27, it showed no difference. And what it meant was the drug was having an effect, but probably not enough of an effect to be curative. This drug ultimately was not developed any further for a variety of reasons, and mTOR remains a questionable target in this disease. Now, I've shown examples of how we can do this and how we can learn as we are developing these drugs. And I'm saying this now in 2021 like it's a new idea. It's not a new idea. Many of you may know Dr. Fred Lang at MD Anderson. He actually published this paper in 2002 talking about all of these different things that we could be doing in our field to better understand whether or not our drugs have true promise in patients with GBM. And so this was an important publication. I think it influenced a lot of people. So in 2020, I led a group that asked the question of, okay, are we actually doing that? Are we really following those recommendations, and are we learning, and what are we learning? So this project I developed via Rano that you may have heard of. And there were a number of collaborators who worked on this with me who are from US, Canada, and Europe. And we went through and looked at all of the different studies. This is the publication. And we looked at all of the different studies over the past 25 years that looked at whether or not the drug was actually getting to the tumor tissue. And we focused in on drugs. We didn't look at biologics. We didn't look at things that are directly delivered. And what we call these in general are window of opportunity trials, that is giving the drug before a planned surgery. Now, there have been hundreds of clinical trials done over the past 25 years. Only 22 of them included tissue-based analysis of drug levels or biological impact. And when we say tissue-based analysis, we're talking about blood or CSF or tumor tissue or tumor-infiltrated brain. So then when we say only 22 of the trials actually looked at something, and then only 50% of those, only half, used the full clinical dose of the studied drug. And when you think about a situation where we're already concerned whether or not these drugs can actually get into the brain and reach the tumor cells, using anything less than the full dose, I don't know how that's going to be informative. And then tissue samples were actually from non-enhancing tumor, and I told earlier that that's really the target. Tissue samples from that target were analyzed in only about 25% of these trials. So it was a great idea to do these kinds of window of opportunity trials. We haven't been doing it. We have to do this. We have to do this if we're going to figure out how to better treat patients with GBM. And part of the problem is the approach that we have taken in our field. This is what we focus on. This is enhancing tumor on an MRI. We see this on the MRI. We say, that's it. That's what we have to treat. But we have a treatment for that. It's a very effective treatment. Here's a post-op MRI showing that that tumor's out, okay? This is the problem. It's this infiltrative area all the way around there. It was there before surgery. It's there after surgery. This is what we need to be treating. That's not what we've been looking at. And that's what we have to focus on. And we can. We have all sorts of tools to be able to do this. This is a study that showed how we can look at these tumors by doing biopsies in different areas, and we'll see different things in different areas. This came out of UCSF. This actually, I found this to be so useful that I've created a standard system that we use now here at Moffitt, where we routinely capture the location at which we take a sample during surgery, whether it's an enhancing tumor or outside of enhancing tumor. And we keep this in a database. We call this geotagging. And now we can link each specific sample that we have to the location of the tumor and ask biological questions. We can use this also to do window of opportunity studies. So I've talked quite a bit about some of the factors that have limited our progress in terms of developing therapeutics that can be given orally or IV. And you know, if that was the only thing that we could do, it would be a pretty short talk, and we'd all leave very frustrated. But there's more that we can do as neurosurgeons, and that is we have unique access to the site of disease, and we might be able to improve on delivery to the area where there's residual tumor. So how can we try to improve delivery of therapeutics to the brain? Well, one way, doesn't involve neurosurgeons, involves medicinal chemists, is to do drug manipulations. That is, change the chemistry of the drugs so that they get into the brain more effectively. And I hope that we can come up with those kinds of drugs. I'd much rather be treated with a pill or an IV than with something more invasive. But we've been trying to do that for a long time. When we change these drugs to make them more brain penetrant, sometimes they have more side effects in the body. So that's an approach that still has a lot of promise, but still needs more development. There have been a number of studies that have looked at trying to couple drugs to molecules that are normally taken up by the brain. That has not worked out so well so far. What about opening the blood-brain barrier? Now, that's been an exciting area of work. Again, over the course of my entire career, we've done a number of ways to try to break open the barrier. It has worked for some types of cancers like primary CNS lymphoma. It has not worked for gliomas. There are some new interesting approaches for doing that using other technologies, but they remain to be proven. So what I want to focus on is how we can directly deliver therapeutics in the brain. Because we have that unique access in the OR to the site of disease, it seems like we should try to go there. And in fact, we have an example of a proof of principle that can work. And that example is the biodegradable drug-eluting wafers referred to or marketed as Gliadel. Now, Gliadel was developed before we had a truly effective chemotherapy for brain tumors. We did use the drug that's in Gliadel, BCNU, but it wasn't so clear that it was effective at the time. And this is now in the 80s and 90s. So Gliadel was created to incorporate BACNU and deliver it directly to the margin of a resection cavity. And in the study that was done with newly diagnosed GBM, it showed a small but convincing survival benefit. Then temozolomide came along, Temodar, which could be given orally, it could be given more frequently. It had a good side effect profile compared to prior drugs, and it showed a much larger benefit, and it had an overlapping mechanism of effect. So combining them didn't make so much sense. Temodar seemed to be a better drug. That's why it's largely replaced Gliadel in the US. But nonetheless, Gliadel was a great proof of principle. It said if we can go directly in there, we may actually have benefit. There are other approaches that have been used: intracavitary infusion, having a liquid in the resection cavity, delivering into CSF, that's the intrathecal term, or into the ventricle in the middle of the brain. And there have been a number of attempts to try to develop therapeutics that way. They haven't worked out so much because the CSF does not circulate within the brain tissue itself very efficiently. And so you don't actually get drug to where you need it. And then there's this thing called the convection enhanced delivery. Okay, so now convection enhanced delivery was developed by a group of biomedical engineers and a neurosurgeon. The neurosurgeon was Ed Oldfield. And this was first reported in 1994. And when you see the word convection, most of you think about the oven that you have in your house, but it doesn't involve heat. Convection just means pressure. And so you're getting heat pressure when you're cooking with convection, but in this case, it's just small positive pressure pushing a drug into tissue. Now, this seems obvious that you should literally pump into tissue, but at the time when this was developed, neurosurgeons like Dr. Oldfield were much more focused on getting getting fluid out of the brain. Swelling from tumors, swelling from other medical conditions was a big problem that they could not manage very effectively. So the idea of actually introducing fluid into the brain just made no sense to anyone, but what he showed was that if you do it slowly enough and over enough time, you can actually get substances into the brain and spread them through the brain. And this schematic gives you an idea of how this really works. Here is a pretend brain with a resection cavity, and these red dots show tumor cells. Remember, we took out in this figure, we took out the solid tumor, and now we have those infiltrated tumor cells, and we want to get our drug to them. Well, we can try injecting. When you do a hand injection like this, you will push drug out a little ways, but that's about as far as it goes. In fact, most of it comes back up along here. Okay, you can put in wafers, and they actually do deliver by diffusion, but unfortunately, diffusion is not a very efficient mechanism. It takes a while to get this far out. And then the drug that's in there, if it degrades quickly, and BCNU does, it's not really active much further than a few millimeters away. Okay, and then you have convection, where you're actively pumping and you can cover a larger area. It's easy to make a schematic like this. I will show you actual data that this works. But first, I'm going to tell you about what can be delivered. And the answer is almost anything. We can give chemotherapies, we can give targeted drugs, we can give oncolytic viruses, other types of viral vectors, engineered toxins, antibodies, which we're going to talk about, and other types of therapeutics. Almost anything, as long as it can be put into a solution that can be delivered into the brain safely. And these are a number of the different agents that have been developed for convection enhanced delivery over the first 2 decades of development. And you can see they have many different types of agents, all delivered within a narrow range of rates, and then the different stages of trials. Ultimately, phase 3 trials, the advanced trials, failed to show survival benefit. And we actually did a failure analysis to try to figure out what went wrong. And as with any failure, there are going to be multiple factors that could be responsible, but we narrowed it on a few that we thought were very important. One was we had no idea where the drug was actually going. We couldn't visualize it in real time. And then the second was in the few cases where we could visualize it, we saw it wasn't going where we wanted because we had non-optimized technology for delivering directly into the brain. So this is an example, and this came from John Sampson of delivery via convection-enhanced delivery. So in this case, it was a targeted toxin that was being delivered, and these are 3 different MRIs done at different time points over the course of delivery. And with this patient, he was able to also deliver a antibody that had a protein that had a radio label. It showed up on a scan. And so what you're seeing is the concentration of that tracer at those different time points, with red being high concentration, blue being low concentration. And you can see you can actually get very nice delivery via that catheter. There's the catheter sticking in there. And you can get very nice delivery over time. And so what's the problem? Well, here's the resection cavity, okay? We want delivery here, we want delivery here, we want delivery here. The reason why this worked here is because this catheter wasn't really ideal for delivering to the brain and had to be put only in places where it had enough of a seal around it to work. That was not over here. That was not over here. And in fact, most catheters that were put in the brain delivered this way when they used the tracer. It just came right back along the catheter and went right over the surface of the brain, never actually got into the brain. That doesn't work. We had to do better than that. The other thing was doing that study was very hard because that imaging agent was very hard to image using the technologies available at that time. And it was very hard to convince the FDA at that time that we needed to be able to give more robust tracers, things like CT dye or MRI contrast agent, along with our study drug. And there were lots of reasons why they didn't like the idea initially, but eventually we were able to. And that has been a game changer. We also knew that the devices that we were using, we were just using tubes that we could use off the shelf because there wasn't a dedicated device. And we knew they were not going to be up to the task because you needed to have a tiny, tiny tip to be able to do this effectively, not the kinds of tubes that we normally use in the brain. So a number of new devices were developed for this purpose. And this is the one that I developed, but there are a number of others that are on the market, and these actually have approvals associated with them. We'll talk a little bit about that. So here's one of those devices. This is the ClearPoint SmartFlow cannula being used with their intraoperative MRI guidance system. Here's a tumor that was being targeted, and here's actually what we infused, and we co-infused gadolinium. So this white here is not gadolinium as it's normally given by IV. We actually co-infused that with the study drug for this study. And this is an intraoperative MRI showing that we actually delivered. Here I am actually doing it. Others have shown the same thing. This is actually in pediatric tumors, diffuse infiltrated pontine glioma. This is work from Mark Suedain at Memorial Sloan Kettering in Cornell, where he was able to deliver an agent that also provides imaging information. So this is actually looking directly at the agent. And this is great work that's ongoing. So we can actually cover these targeted areas. But let's come back to GBM. And I'll show you again this figure that I showed where we can take out the tumor, but now we have this large volume here to treat. It's not a small volume, it's a large volume. We actually did some work to characterize how large that volume was, and the median, that is 50% with more, 50% with less, was about 37 cc's, some as high as 170 cc's. And when we look at that device that I showed you that we have to use in the intraoperative MRI, the most we could probably ever deliver over 6 hours is 3.5 cc's. That's not enough. That's not going to work. That's not going to cover these kinds of volumes. So how do we do that? Well, this is where my background in biomedical engineering came to help with this project. So I worked with a group of actual practicing biomedical engineers who were device developers. They had experience developing medical devices. And so we set these parameters. We wanted reliable high volume drug delivery that could be used both in the OR and out of the OR. Time is our ally. The longer we can deliver, the greater the amount of brain we can cover. And of course, it had to be resistant to backflow. Things had to move forward. They had to be biocompatible, drug compatible, and MRI compatible materials. and it could be placed using conventional equipment. This is not something that should only be done at a very, very specialized center, something that ultimately we want lots of people to be able to do. And this is what we came up with, this multi-port catheter for which we have multiple patents. And we found a manufacturer, Parker Hannifin, who actually makes the device for us. And we formed a company called Infusion that is doing the commercial development. But this has 4 independent microcatheters, and we can place this where we want in the brain. Now, making a device was not enough for me. I wanted to prove that it actually worked. And that was this initial study that we did using topotecan, which Jeff Bruce at Columbia had been using and published on. And we also co-infused gadolinium. This just shows some of the early work in the OR and with a patient who went for 48 hours with these catheters in and having the topotecan being infused directly. And this shows some of the results from our early study. We learned one important lesson, and that is if we put a catheter in the tumor infiltrated brain, we get much better delivery than if we put it into the enhancing tumor. Okay, and that's because the ability of the drug to get out into the brain depends on how quickly it's taken away by the bloodstream. In an enhancing tumor, you have a broken blood-brain barrier. The drug ends up just being washed away, whereas in the infiltrative brain, that part that's behind an intact barrier, now the blood-brain barrier is your friend. It keeps the drug in there. And so we did this. We showed that actually it's better just to do it in the tumor infiltrating brain if you want to have a therapeutic effect. This was a Journal of Neurosurgery publication. But then what we did was we changed the protocol. We took out the enhancing tumor. This is actually a resection cavity. We put 2 catheters in, and now we're starting to cover very large volumes of surrounding brain. Now we're talking about actually getting out there where the infiltrating tumor is. And that was the exciting development from this. Now what we're actually doing at Moffitt, now that I moved down here, is we're doing another protocol. We're saying, can we actually accomplish both? Can we cover both the enhancing tumor and the tumor infiltrated brain? And this is actually one of the patients in this protocol where we put one catheter into enhancing tumor. This is the MRI with IV gadolinium. We started pumping at a higher rate, and now we're getting both the tumor and the surrounding brain just with that. So this is, again, a really exciting development. And so with that, we're gonna transition over to Anne Marie, who's gonna talk about the exciting drug that she's developing that really is ideal for treating those tumor cells that we can't remove surgically. So Anne Marie.
Thanks, Mike. So yeah, I'm gonna, let's see, okay, no more echo, perfect. So I'm gonna, as Mike said, talk more specifically about our clinical trial that employs a lot of those important principles that Dr. Vogelbaum discussed. But first, I wanted to just talk a little bit about the preclinical potential and obviously what we saw in this therapeutic that prompted us to develop this and now test it in humans. So the first thing that we did when we were thinking about how to address the problem of cancer, and more specifically glioblastoma, was to rethink the current approach. So just at a very high level, you know, there has been a trend in oncology away from the chemotherapeutic agents that we know that indiscriminately attack both the cancer cells and the cells in our body, and towards targeted agents. But there's these targeted therapies that address these single biological processes driving cancer and growth and progression still have some drawbacks. And that's what I'm showing on the left of this screen, is that when we only address a single biological process like bevacizumab does in terms of preventing new blood vessel formation, these cancer cells are really smart and adaptable, and they actually will rapidly and readily hijack other biological processes to their survival benefit and ultimately become resistant to these therapies. So with this in mind, we decided that what we had to find was something so central to cancer development, growth, and progression that we blocked multiple of these biological processes simultaneously. And go to the next slide, please, Mike. So if you're trying to find a new approach, you have to start thinking outside of the box. So as I said, for decades, we've been looking at these cytotoxic therapies, and we've been really focused on the cancer cell itself. So when I mean out of the box, we started looking outside just the malignant cancer cell. And we started looking at the tumor microenvironment. And what we pretty quickly discovered is that just like human beings, cancer cells are constantly influenced by their environment. So we thought, well, what if we can potentially, and sorry, I'm going to back up. One more important thing is that this microenvironment we learned actually is constantly in communication with the cancer cells. So, and when it communicates with them, it's really supporting their mission. And so cancer cells' fundamental mission is to continue to grow, to invade, and then and to progress. And so what if we could quarantine the cancer cell from those growth promoting cancer signals and cut it from its lifelines? So we had to find what is the key receptor that actually is integrating this communication. And that is where CD29 enters. And so the therapeutic that we're developing is an antibody therapy against CD29. And as Dr. Vogelbaum said earlier, as an early stage company, we have named it OS2966, which is our internal reference. So that's our anti-CD29 therapeutic. And so, yeah, perfect. And so how does CD29 actually work? And so what you'll see is that when it cuts off these signals, it's actually involved in every single step of cancer growth, invasion, and resistance to therapy. So if you look at this image, which I know you can't because I can't you can't see my mouse as Mike is sharing, but up top on number 1, so CD29 actually leads, when it gets this information from the surrounding microenvironment, it transduces that information into cell signals. And these signals within the cell cause cancer cells to grow, divide, and proliferate. But it does even much more than that. So it actually changes the shape of the cancer cells and the mechanical properties of them to let them escape the normal confines of their structure and environment. And then even an additional step that it does is it actually affects cancer cell motility and allows it to escape from its primary location, enter the bloodstream, and spread. And then again, so now you have the cancer has grown, it's invaded the tissue, it's spreading through the bloodstream, and now it has to find a way to survive in its distal or foreign environment. And so it helps set that up as well. And finally, CD29 has been linked directly to drug resistance. So this is a really, really central cancer driver. And our goal was, again, to bring this forward into the clinic. But before that, we did a lot of preclinical studies. And so the next slide, I won't go through the data, but what this shows is that we have studied OS2966 in several cancer indications. Most exciting to obviously our audience today is in therapy-resistant glioblastoma, but the data looked really good across all of these therapy-resistant cancers. And so we set out to design a clinical trial first in glioblastoma, and we took into account a lot of those key principles that Dr. Vogelbaum discussed. And so I'm excited to tell you a little bit more about the trial specifically. You can go to the next slide, please, Mike. All right, perfect. So our phase 1 trial is currently enrolling at Moffitt Cancer Center, obviously in collaboration with Dr. Vogelbaum and his neurosurgical team. We are employing the technique of CED, and we are using the Infusion Cleveland multi-port catheter. Also important to the trial, before I talk about more of the logistics and details, is that we have optimized the trial design to collect tissue both before and after treatment, so we can really understand if the drug works through its intended mechanism, and also study more of those, its pharmacokinetics, so how the body responds to the drug as well. In terms of what the trial looks like, right now it's a single site. We are dose escalating. Obviously, we start off at a low dose and we increase through our protocol to explore up to what we hope to be the maximum or the optimal biological dose. In terms of the goals, so a phase 1 study is typically designed to test safety, but we wanted to get as much information out of this study as possible. So we're looking at safety, we're looking at dosing, we're looking at specifically how does the drug distribute between that contrast enhancing brain and the tumor infiltrating brain that Dr. Vogelbaum talked about, zone 1 and zone 2, and how does it actually spread so that we understand the delivery technique with the Infusion Cleveland multi-port catheter. We're also going to be looking at efficacy. So is it working? When we look at imaging, are we seeing that the disease is progressing? Are we seeing that we're getting stable or some response? And then importantly, again, we're gonna look at the tissue, which I'll talk about a little bit more, and see, does, I've told you at this really high level how important CD29 is for cancer, and it affects all these signaling pathways. But this is going to tell us which signaling pathways does it really touch on, and are certain patients, in certain patient populations, can we correlate that and see that some patients may in fact benefit more from OS-2966 than others. And so in terms of selection, obviously every clinical trial has a lot of selection criteria, but some of the more important ones are that the patients have to have a recurrence or progression of their disease. They've had to have been treated with Temodar or temozolomide and radiation therapy, and they need to require a resection because we're doing this we're administering the drug in the perioperative, the perioperative manner. The tumor, there's certain size and characteristics about the tumor in terms of the protocol, but I think the most important thing is requiring a clinically indicated surgical resection. We have a treat, resect, treat design. The reason we do that is to optimize to collect tissue, and then again also to understand how the drug distributes in these two distinct areas. In terms of other things, or dose escalation, these are things I'm proud of. From your perspective, they may not be as exciting, but we have a concentration-based dose escalation. So most drugs, when you're designing your dosing, you think about the patient's overall body weight, and that's how you come up with your dose. But when we're delivering something directly to the brain, we really didn't make sense. So we had to come up with a different metric of how to deliver this, which makes more sense. Lastly, I think what I'm proud of about the study is we've really married, the viewpoint of the neurosurgeon, the drug developer, and the patient. And so I think that to the best of our ability, we've made it friendly to all of those parties. Okay, perfect. And then taking a little bit of a deeper look into the design. So we'll start at the very left of this graphic. So after enrollment criteria is met, the study is carried out in two distinct parts. So in the first part of this study, the first thing we do is take a biopsy of the tumor, and that's how we get that pre-infusion tissue, and we're also able to confirm recurrence. Next, the Infusion Cleveland multi-port catheter is placed, and in the first part, we're actually administering the therapeutic via CED to that enhancing part of the tumor, or zone 1, and with the plan of coming back and resecting it. So per the protocol, 1 to 10 days later, right now what we've been doing, we've treated 2 patients on this protocol so far, and they've been spaced out 2 days. But we bring them back to the operating room in study part 2 for their planned clinically indicated surgical resection. We get that tissue and we're able to use it for a lot of different important studies, which I'll talk about briefly. We're almost out of time. And then we place the 2 additional catheters in the tumor infiltrated brain, zone 2. The patient is then woken up from anesthesia, and they're infused at the bedside over 4 hours. And then the follow-up period begins. So there's initial follow-up 15 to 21 days after the surgery that coincides with suture removal for safety, and then every 8 weeks. And that's when we start looking at those MRIs to measure response. And I think this is my last slide. So I just wanted to mention that that's just a duplicate, Mike, so we can go on. Before Dr. Vogelbaum talks, that I'm really, I think the tissue part of the study is really exciting. It's very important to me that we're seeing things at that level, biological activity. And if we're not seeing the drug binding, that we know we're able to course correct. And someone asked me a good question when Dr. Vogelbaum was having technical issues. And so we're really, why do these early trials work and the later phase ones don't? And that's what we're really trying to prevent by getting all this information. So back to you, Mike.
Well, I think that's great. I'm just going to acknowledge a number of people who've been involved in helping to get this trial off the ground. This is the team at Moffitt. and a number of our funding agencies who've helped to develop this. Of course, Musella Foundation, as I think you want to acknowledge, has been so helpful for this specific trial, getting this up. Ultimately, I think that we're all on the same page here. We want to stop cancer in your head, and if we're going to do that, we have to deliver. And so I'll just leave you with a few images from my new hometown.
Thank you. It's very, very interesting. We just have a few questions. A lot of times, most drugs that are used for brain tumors are given over a period of 6 months to a year, whatever. And you seem to only be doing it for like 1 to 3 days. Is there any way to leave the catheters in and just give treatments like every month for a long period of time?
So I think that's a great question. And you know, the schedules that are used for different types of drugs are very dependent on a number of different factors. In particular, how the drug or biologic works. So there are some things that do require continuous treatment, sometimes even lifetime. When you think about signal transduction inhibitors for some diseases or other diseases, immunosuppressants. But you know, in cancer, it can vary. It depends upon the strength of the effect. Ideally, we do want to be able to do that, be able to do a chronic delivery of this or any other therapeutic, or a chronic intermittent delivery, and have a system left in place. That's where you get into the challenge of doing device development. It's very expensive. These are delivery devices. They're not therapeutic devices in themselves. And so it really comes down to who's going to pay for it, because I can tell you that I have lots of ideas. I have some patents filed on some chronic delivery devices. It's a question of raising the funds to be able to do all the work before I could put one of these into the first patient. And that's really what we need to do to get there. Yes, I think in the end we're going to need to have long-term implantable devices, but it's going to take. We have to take it one step at a time to get there. With a drug like this one, with the OncoSynergy drug, you know, if we're seeing therapeutic effect, if it looks like it's working, the idea of then doing multiple administrations over time, even with a small surgical procedure, really takes us about 20 minutes to put the catheter in. You know, with a small surgical procedure, if that's what we need to do initially to get to that point, I'd love to be there where we have something that's actually working and helping people.
Okay. Is CED practical in general? Like, can it be done at any hospital or only at the major centers?
Right now, it is really just done in an investigational setting. So when you talk about that, you need to have, you know, a highly coordinated team, highly skilled team that understands how to do clinical trials, that is going to do advanced clinical trials. That's now. But like with any technology, once it really reaches the point where it shows efficacy, it becomes approved, then the next step is to get it out to as many different hospitals as is feasible for that kind of technology.
Okay. You mentioned a whole bunch of failed CED trials that use the older technology. Do you think it would be worthwhile to even try those again using the current treatments?
So yes, absolutely. I think that would be very interesting to do. Now you're getting into again back into some of the economics of it. Ultimately, you know, Anne-Marie can tell you more about how much it costs to develop a new drug. And many of those drugs now, the old ones, may not have a patent life anymore.
Oh, that's
so
good.
if there's no return, you know, on investment, then who's going to pay the millions of dollars for those trials?
Well, it's funny that you
Right.
mentioned that, because on my talk in about 4 weeks, we're going to address that issue directly, where we're going to try to
Right.
make it much cheaper to develop drugs, to the point where the nonprofits like my organization can actually develop a drug, and especially if it has no patent on it, we would pay for all that stuff and get a drug that, once it's developed, we could sell for relatively cheap and just get
Right.
cures.
Yeah, I think it'd be fantastic. There are actually some efforts, though, to take those same principles and those drugs and improve upon them in a way that generates new patents, which is great, because then we could also use some of the existing approaches to do that. Excuse me. So some of those drugs are actually being improved upon,
Okay.
and that's actually very exciting.
Yeah, let me see. You mentioned Gliadel. Would it make sense to use Gliadel with different drugs other than BCNU, or is it just too limited?
So, many of you may be aware that Dr. Henry Brem developed Gliadel, and his laboratory has looked at a wide variety of different chemotherapies that could be incorporated with it. In the end, though, the mechanism of diffusion, that mechanism is not very efficient and is not going to get as far out into the surrounding infiltrated brain as we would like.
Okay. Another question, this one from Facebook. Do catheters lead to infection? Is that one of the reasons you only leave this in for 3 days?
Right, that's exactly the risk. That's exactly right. So anything that penetrates the scalp and is external, you have a limited time window. That may be as long as a week, but we're now dealing with a situation where we want to try to be more conservative than that because the infections can be very very debilitating. So we limit it. And actually, what I found was with the infusion rates we were using, we could actually cut it down to only 2 days, where we really hit peak effect. In the end, I'd love to see a fully implantable system that we could develop in the U.S. For our patients, and there are a number of ideas and approaches I'm trying to work on to do that, but it's no one's really been banging down my door with money yet, nor anyone else who's been in this field.
Hopefully we will soon. What are your thoughts on gamma tiles?
So, that speaks to the point I made earlier about brachytherapy. That is what we refer to as short distance radiotherapy. For certain situations, it actually might make sense, for example, for brain metastases. We know we have to use radiation in the resection cavity. I think that there could be a lot of interest there. The data I've seen so far to date show that it's safe to use them in general. I have not seen convincing efficacy for glioblastoma in particular. Okay, there have been single arm studies that have shown that it's safe, and when you compare to historical control, sure, it looks great, but you need more than that to really be convincing. We've been led down many false paths.
Okay. You mentioned opening up the blood-brain barrier. There's a question about two technologies. One was focused ultrasound.
Right.
What are your thoughts on that?
So I alluded to those technologies. There's
Yeah.
lit and focused ultrasound that are showing some very interesting data with respect to opening the barrier to the point where some contrast gets in. Now, is that enough to get drug in? And for how long does the drug stay there? And is it enough to change, to affect the tumor? Those are the studies that still have to be done. And I'm actually quite interested in it and looking at opportunities to engage in that kind of work. Because like I said, anything that's going to work, I'm a fan of. We just need to study it carefully. It's really easy to make promises and to be all excited, but we have to do the hard work to really figure out whether or not it's actually working and how to make it work better.
Yeah, something you said made me think. If you do focused ultrasound to open up the blood-brain barrier, pump the drug in, and then close the blood-brain barrier, that'll actually trap the drug inside, correct?
So when you say pump the drug,
well, I mean, just deliver the drug, IV or whatever.
So the idea behind using focused ultrasound to open the barrier is to give the drug systemically and let it get in that way.
Right.
When it closes, and presumably it stays in. But it's not entirely clear how much you get in. Okay. There are other mechanisms of the blood-brain barrier besides the tight junctions in the blood vessels. There are active pumps that push things out. And so it's not clear how much you're actually getting in. And that's what we have to look at. We have to look at it in the tumor-infiltrated brain, not in the enhancing tumor. Those are studies I think that are very important to do.
Okay, another question about that is, what about super selective intra-arterial delivery of drugs?
Yeah, I'm glad you mentioned that because then I could talk about one of my new faculty members, Dr. Andre Bierferlin, who is both a skull-based surgeon, does endonasal, endoscopic, skull-based work, open skull-based, but he also is trained in endovascular neurosurgery. And that's his research interest, is to try doing super selective intraarterial chemotherapy. That's a program that we're going to be standing up over the next few years. Again, it's I want to see the proof. I want to see it in the tissue that it's actually getting there more effectively and acting on the tumor tissue in a way that is likely to provide survival benefit.
Okay, question for Anne Marie. Who would be the perfect patient for your clinical trial? Like what circumstances?
That's a great question. So obviously, one that I guess the easy answer is one that meets the protocol requirements. And those are the most important. What we've seen, and Dr. Vogelbaum and I through the screening process, is that it's really the tumor location and size. So in a phase 1 trial, we're being more conservative. So we have these criteria around where the tumor can be located. We want to make sure it's not close to the ventricle, things like that. So a perfect patient would be a patient who meets those requirements, and so specifically needs a resection. Yeah, that's a tough question. But yeah, the tumor requirements are the toughest. I don't know, Mike, if you have any other highlights about what the perfect patient would be. One that's really nice to you? I'm just teasing.
Yeah, no, I'll tell you, it's I'm so grateful that people will consider doing these trials. Yes, in the end, this is something we're offering because we want to try to come up with something that's going to be more effective than what we have. But it's hard. I mean, it's hard to contemplate the idea of going on to an investigational trial. Folks really would much prefer to have someone tell them, I have something that's really going to work for you, and then just do it. But in all honesty, that doesn't exist, and I think that it's really incredible that people will trust us to help them, better than we have in the past, and so it's really folks who understand that, and we ultimately develop a partnership in these, and a lot of patients I've treated on clinical trials have been on more than one trial, because they understand this is hope for them, and we're doing everything we can to make it as safe and as effective as we can. But that's really, it's really a relationship is what it comes down to.
Okay. Is it feasible to have a patient who has to travel come to you, or should it be a local person?
For this trial, I think it is feasible. The only challenge is in the follow-up, but that's not that frequent, and there are lots of flights to Florida, so it's not so bad. So it's better than some of the trials that require someone being here for weeks and weeks on end, or having to come every 2 weeks for a treatment. This one actually is a bit easier. It does require that period of time with the surgery and then the initial recovery, but after that, then it's much more routine follow-up than anything else.
Okay, if somebody's interested in the trial, who do they contact?
They contact me and my office, or they can contact Anne Marie. Anne Marie's been fantastic at talking to people about the trial and then in bringing them over. So either way.
Okay, just send me the contact information. I'll put it on the recording. And the last question, I think, let me see, is, do you think that there's going to be any new treatments approved by the FDA in the next year or so?
For any glioma.
For any glioma?
Yeah, I would say that in the sense of something truly new, boy, I'll tell you, that's a hard one. I'm not sure, unless there's some accelerated approval for one of the drugs that are more brain penetrant. Perhaps in the setting of brain metastasis, we might see that, but for gliomas, I think it's still a little bit early to be quite so optimistic, but who knows? I mean, it's a little challenging to make that kind of prediction. We've seen some exciting results from the viral gene therapy trial at University of Alabama for pediatric glioma. I think that's very exciting. Could that lead to an approval? Maybe a little bit early, but I think one of the things I've learned about the FDA, it's always easy to sort of point fingers at the FDA and say that they're in the way. I'd say actually, when we look at the track record of what's actually worked in this disease, where there's convincing evidence, and the way that they've worked with me, I know on a number of different trials, I think they've been bending over backwards to try to help us, try to find something that's going to be effective. And to me, the Avastin approval just showed that they would bend so far as to take something that was really just a radiographic effect and make it available to patients. That was going pretty far.
I hate to say, but
trying to help us.
I think that scared them and made them a little more conservative.
Yeah, maybe a bit, but I think they've been trying to help us. I think that their track record says that they really do. They understand. And they do want to help us.
Okay.
We have to come up with something better.
True. I was thinking along the lines of like DC-Vax or ONC201. Those are hit or miss. That's the last question. So I would love to thank you both for joining us. It was very informative. And I'm going to edit this video and put it back on the website with your contact information. So if anybody's interested in the trial, they could contact you. Thank you. Wonderful. And we'll see you guys in 2 weeks. Well, not you guys, but the people watching.
Thanks,
Thank you very much.
Thanks so much, Al.
Bye-bye.
Bye.
Bye.